Okay, good evening. Welcome to our Unit 3 BIO205 exam review. Thank you for joining me at this late hour. I do appreciate it. For those that cannot attend, you can watch this session later and then message me with any questions if you have them. Do I already have a question? Oh, hi, Fahima. Okay, so I'll go ahead and get started. I won't take up too much of your time. It's kind of holiday season. Everyone's got a bunch of stuff to do to prepare for um, either Thanksgiving or Black Friday. So I'll go ahead and roll into it. So quick overview, I'm going to talk about essentially reviewing the first line, second line, and third line defense. And I'll kind of show you two kind of practice cases to kind of show you how the questions could be worded on for this exam. Again, most of this chapter, I think all of this chapter was specific to the first, second, and third line defense. So understanding how to differentiate between the type of infection and then how the immune system will response will be very key to getting a good grade on this exam. Some quick details. So this exam will be done online. The exam opens Wednesday at noon after the Wednesday lab completes, and it'll be open until Saturday night. If you want to, or if you need to take it before Wednesday, you are able to take it with Dr. Cotter tomorrow, Tuesday, between 10:30 and 11:30 or 10:30 a.m. to 11:30 p.m. But you do have to message her so that she can confirm and be there in the lab DB123. Um, so if you do want to have that option to just get it knocked out of the way, please message her. Otherwise, you'll have a couple days to review, prepare yourself, take the test. If you don't want to take it on Wednesday and don't want to interfere with the holiday, you still have your Thursday, Friday off, and then you can take the exam on Saturday. This will use Respondus with audio and webcam enabled. So make sure you follow the general rules. Try to pick a quiet location, get reliable internet. Make sure you do your screening or your swiping with the camera around your locations. Make sure nothing really stands out like pieces of paper or your phone or tablets or whatever, just to not give the impression that you might be cheating on the exam. Protect yourself by not making it questionable what you're doing on this exam. Okay, so first line of defense. So essentially, the first line of defense, the main purpose is to keep microbes out. So it's the first thing that we have available to prevent entry of microbial infections or different types of pathogens. You can organize this different ways. The way I decided to break it down was between physical and chemical barriers, because this is most likely how you're going to get asked some of your questions. And I also like to break it down like this between different locations, such as the skin, the respiratory system, your eyes, your GI tract, or reproductive. Because for the most part, the first line of defense, because it's non-specific, is very similar. So on the physical barriers, your skin, you have your epidermis and your dermis. For the respiratory tract, you've got mucous membranes, mucus, cilia, also the act of coughing and sneezing. Eyes, primarily your blinking is your physical barrier. And then you also have like eyelids and things like that. GI tract, again, mucous membranes, mucus. This also includes vomiting and diarrhea. And the reproductive tract primarily uses mucus, mucous membranes, and cilia. Um, Most likely, the questions are going to be revolving around the respiratory GI tract or reproductive system. So you can already tell, like if you have mucus, if I ask you for three physical barriers for first line defense for respiratory, GI tract, and reproductive, at a minimum, you should be putting mucus, membranes, and mucus. And just like in page one of your study guide, you're going to be asked to give a quick description for some of the questions. But for the most part, mucus and mucous membranes should be first line physical barrier for almost any case you're given. And then after that, it's just kind of thinking it through like, okay, if it's respiratory, I'm sneezing, I'm coughing, GI tract, I'm vomiting, or I'm going to have diarrhea. And reproductive tract also just has cilia involved. For chemical barriers, the skin has an acidic pH. It's very salty. And then you also have some of your oils, your sebum or sebaceous oils, which are bactericidal or anticidal, which helps kill bacteria. Respiratory tract, anytime you see something that says mucus, you should also associate lysozymes, and lactoferrins for the chemical barriers. Um, there are things like gastroferritins or gastroferrins, like the GI tract message. Talking to Dr. Cotter today, if you have mucous membranes and you put lactoferrins for GI tract, she'll still give you credit. So if you have a physical barrier of mucus and mucous membranes, and if I'm telling you that most likely you'll have reproductive GI tract or respiratory, then lysozymes and lactoferrins should be almost guaranteed to be two of the chemical barriers that you list down. And if you get asked for like a third or a fourth, you can then do the very specific ones like the acidic pH or hydrochloric acid in the GI tract, or the acidic pH in the respiratory tract. So a lot of your linings are actually very acidic, besides the small intestine. So again, you can use acidic pH. You can see that I've listed that for three of the five locations in the body where microbes might enter. Resident microbes, again, you most likely get asked a question about either one or two resident microbes you might find in this specific system that the case study is revolving around. So just memorize two of them per location. Some of the ones that you should already know by this point: Staph aureus, Staph epidermidis, Escherichia coli. Things like in the vagina, you should also know things like Candida albicans. Um, and the biggest thing with these questions is you might be asked, okay, list a resident microbe and give me two ways that resident microbe protect the host. Very similar to the chapter 15-16 case study. You should see something very similar on this exam as well. And just remember, to recap, that the resident microbes help block attachment of pathogens, help compete for nutrients, and are also give off antibiotics to inhibit the growth of other microbes. Okay, second line of defense. This is usually where it starts getting a little, I'll say, not challenging, but where I see students start missing points is usually the second line of defense. Um, this is from page 14 of your handout. And biggest things to highlight is if the question asks you for two second line immune cells involved in whatever infection, bacterial, viral, it doesn't matter. You should always put neutrophils and macrophages. Again, this seems pretty simple, but I feel like every semester there's always a good handful of students that forgets to list one or two of these. The question could ask you which second line immune cells are always involved in the response, or you might get asked, hey, you have a respiratory infection. What second line immune cells do you expect to be here? And neutrophils and macrophages should be involved every single time. Very similar to the chapter 15 case study, if you recall that we had you list, I believe it was three second line cells, and then three chemicals, and then three processes. The question is going to be very similar, and you'll see it in the practice case I have for you all later. But if you list macrophages as your second line cell, you should be able to list that the macrophage releases digestive enzymes, can release interleukin 1 or interleukin 6, and also releases the complement proteins, and what each of those complement proteins does. So know the second line cell, the chemicals, and then the processes that are involved with the chemicals that you listed. Other things to remember, things like basophils and mast cells, those are involved in allergic responses. So basophils are any time an allergy makes its way into the blood. And then most likely the question you might have is a mast cell allergic response, where you take something internally to the tissues, like through the respiratory tract. And then eosinophils are also involved in parasitic infections. Um, for the Monday group, we worked through those practice cases on page 9, and parasitic infection was one of them. Just remember, if it has parasites in it, the eosinophils should always be involved. And they do the same thing, they just are involved in phagocytosis of parasites. And third line of defense. So this is where I really see the divergence in the grades. If you do study your exam, what's going to make or break the exam for you is going to be the third line defense. I broke it down in a certain way here to maybe help you memorize it a little bit better. Page 24 of the handout also has a breakdown like this. I just reworded it a little bit differently. So that maybe, hopefully, either this or page 24 or the way Dr. Cotter explained it with her visuals with having students be certain cells. Hopefully one of those clicks for you so that you can answer this question properly. So third line defense are involved with directed or specific responses to the infection. So they will, if you have COVID-19, it's going to directly create a defense against the COVID-19 virus or a Staphylococcus aureus infection, whatever. Um, the way you want to differentiate these first is determine if the infection is inside of our cells or outside of the cells. And I put examples like the dermis or blood vessels or in your respiratory tract or in your GI tract. Um, if the infection or the pathogen just hangs out outside of your cells, it's an outside attack. If the infection goes inside of our cell, which is primarily viruses or cancer, and in our case, most likely viruses, um, then that's going to involve one type of response. Everything else is going to involve the other type of response. So if the infection is inside, like a virus, you could see wording such as an intracellular response or a cell-mediated response. Kind of like Dr. Cotter said, you are number one. So I put examples, again, viral infections. So the macrophages are going to take this information. They're going to take it to the Th1. So again, you are number one. So Th1, CD4 plus helper T cells. And I put that big long word. You don't have to say helper T cells. You don't necessarily always have to say CD4 plus T cells, but you should be able to say that it's a Th1 helper T cell or Th1 CD4 plus T cell. Um, so macrophage will talk to the Th1 CD4 plus helper T cell using the CD4 plus receptor. This will activate the CD8 plus cytotoxic T cells and memory T cells. And the T cells, because our T cells will use T cell receptors or TCRs to screen each of our cells. So kind of like the TSA check in your passport. And then cytotoxic T cells will destroy virally infected cells using perforins and granzymes. That description is basically bare minimum of what you should be able to list out for the cases. And you can kind of see how this one statement here could probably be worth something along the lines of like 8 to 12 points on a question. Just that it asks you which helper T cell are you reporting to? What cells are activated by that helper T cell? What receptor will those cells use? And then how will those T cells destroy or attack the infection? And again, I have two different examples after this to kind of show you how we could break this down. And then the other one is outside the cell, which is basically everything else. So you can see this being written as an extracellular or a humoral response, and that is priority number two. So that includes fungal, bacterial, parasites, and allergies. So the macrophage still involved with this will report to the Th2 helper T cell using the CD4 plus receptor. This will activate plasma B cells and memory B cells, and then those plasma B cells will use B cell receptors or BCRs to look for the invading microbe. Once it finds it, the plasma B cells will secrete antibodies to surround the microbe and do four things: agglutinate, neutralize, opsonize, and then perform complement fixation. If you guys don't remember what these terms mean, agglutinate is just clumping. So antibodies are like Lego pieces and they can attach to different microbes and then they can attach to each other. So it makes a much larger clump of antibody and bacteria, for example. And it's just a much larger chunk of stuff that the macrophages will want to actually eat up first. Neutralization, the antibodies can attach to places where are the bacteria would want to attach itself to like our respiratory tract. And so by blocking those attachment points off, that bacteria, while it's inside your body, it can't actually attach and start breaking down our mucous membranes to enter. It just stays there and it's basically neutralized. Optimize is the seasoning of the salt. So we all like steak, we all like chicken, or we all like different types of foods. And when something doesn't taste well, or if we want it to taste better, we add things like seasonings or dressings. Optimization is just the antibodies adding seasonings to the bacteria to make them more attractive for the macrophages to then come eat, so they'll eat them up faster. And then complement fixation, that involves your complement proteins. It actually helps your complement proteins do their jobs, primarily the MAC attack helps make a bunch of holes in the parasites or the bacteria to make them break down easier. And once the inside cell parts make it outside, our body can easily eat those up a lot faster. I'm not going to cover so much the antibody types here, but I do recommend you just go over them. Biggest things to note, IgM is the first antibody that's made, and it's the largest. IgG is the second antibody that's created, and it can travel anywhere in the body. It's small enough and can cross into the placenta. IgA is involved in mucosal infections. So anytime you have a mucous membrane, like your respiratory tract or your reproductive tract, IgA will be involved. And then during an allergic response, IgE will be created instead of IgG. Okay, so how to tackle a case study. So I have two examples. One is a bacterial infection, and the other is a viral infection. Um, and so the way I want you to break this down is as you're reading this question, and don't skip ahead because you might miss key information. I would try to go in order for the cases. That way you get all the information you need and you can work your way down. So I want you to ask yourself 5 questions. First is, okay, where is my infection and what pathogen is causing it? So the example I have here is a bacterial infection in the vaginal canal. So the first question or the first thing you want to ask yourself after that is what first line chemicals and physical barriers are involved. So physical barriers, like I mentioned, mucous membranes, mucus and cilia. And then whenever I say mucus, you should have the chemicals lysozymes and lactoferrins involved. And then you also have an acidic pH in the vagina. That right there is probably worth 4 points. So on my practice case, that's worth 4 points. So I'm going to want you to give me 2 physical and 2 chemicals for this bacterial infection. So in question 2, that's 4 points right there. The next question is going to ask you, hey, do I have any resident microbes in this area? I gave you 2 here, Candida albicans and Lactobacillus acidophilus. I recommend you have at least 2. That way, if you're taking the test and you forget one of them, you might remember the other one and either get partial credit because it's asking for 2 of them, or you get full credit because it might just be asking for 1 resident. So in this case, I'm asking for 1. That's 5 points right there. Then the next question is, okay, now what second line cells are involved? What chemicals are involved? And then what processes are involved on the second line? So, like I mentioned, macrophages and neutrophils are always involved. Those should always be listed in your cases, in your practice cases on page 9, and in your cases on the exam. Macrophages and neutrophils, what do they do or what chemicals do they give off? Well, I have digestive enzymes for both macrophages and neutrophils. I also have interleukin 1, interleukin 6, and then the complement proteins C3b, C3a, and 5a, and C5 through C9. And then what processes are involved. I'm going to have phagocytosis, fever, inflammation, and the membrane attack complex. Um, page 24, or sorry, page 14, if you're like, oh, what do all these do? This breaks it down really well. That's why I wanted to share this one. So So say the question asks you for two second line cells, two chemicals, and two processes. That's already 6 points right there. So far my question is already worth 11 out of the 80 points. And then the one that, like I said, always gets people is the fifth part. Okay, now on the third line side, is the infection inside or outside of your cells? So bacteria, that is an outside of the cell infection, which means I need to be involved in an extracellular or a humoral response. So that will involve the Th2 CD4 plus helper T cell, or your general, as Dr. Carter likes to say. That will involve your plasma and memory B cells, and that will involve the antibodies, specifically IgM, IgG, and IgA. Getting rid of like the bacterial outside extracellular response. One question might be for the Th2 portion. The second question might ask you if it's T cells or B cells, and you'll say the B cells. And then the third question will say, what is involved, either perforins, granzymes, or antibodies, and you have antibodies. Right there, that's another, let's say, 3 to 5 points. So this one case study that I'm just kind of working through is already worth what I say. I said 4, 5, 11, and let's be nice and say 4 points. So that's 15. So that's 15 out of your 80 points on just this one case study. And you're most likely going to have, I'd say, two or three like this. If I was to give you the exam, I'd probably give you one or two of the outside infections, and I would definitely give you two questions to be able to show me whether you can differentiate between a Th2 and a Th1 response, because students missed that on this last case study and typically tend to miss those. So to see if you actually studied this, you want to be able to say, can this student differentiate between a Th2 and a Th1 response? And tell me if it's plasma B cells or cytotoxic T cells, and then what chemicals are involved to protect you from that. Okay, so I'm just going to go through another one here since I'm already about 25 minutes into it, and then I'll open it up for questions and then wrap it up. So a question that you might have is, hey, your patient comes in and they're complaining about fever, chills, headaches, and they've been coughing and sneezing since yesterday. You take a throat culture and the lab returns the presence of COVID-19. So again, question one, what is your infection? What is causing it? Or sorry, where is your infection? What is causing it? Like I said, COVID-19 is a virus, and where is it happening? Since I'm coughing and sneezing, most likely it's happening in the respiratory tract. So again, now go back to your first line. What are my physical barriers? I have mucous membranes, mucus, I have cilia, coughing and sneezing. You most likely will need to ask, you most likely will need to add all five, but you might be asked for two or three. And then chemicals involved, lysozymes and lactoferrins. Again, mucus, think lysozymes and lactoferrins. Any residents in the respiratory tract, again, our big one, Staphylococcus aureus. You can also add Staphylococcus epidermidis, and then I believe our list has another one listed. I don't necessarily care about that. Just get two down that you can remember. That way, if you forget one, you can list off the other. All right, go back. What are the second line cells, chemicals, and processes involved? Again, macrophages and neutrophils should always be listed because they're involved in every immune response. In this case, because it's a tissue, we're going to also have mast cells involved, and those are going to release histamine. And then the same chemicals, digestive enzymes, interleukin 1, interleukin 6, and then the complement proteins are involved, and then they are all involved in phagocytosis, fever, inflammation, and the MAC attack. And then the big one is the infection inside or outside your cells. Viruses infect inside the cells. So that is an intracellular or cell-mediated response, which means I need a Th1 helper T cell. I need cytotoxic and memory T cells, and I will have perforins and granzymes involved in the immune response. All right, that's really all I had for that. Again, these two examples, like I would study these, make sure that they make sense to you. Like I said, for the Wednesday group, On Wednesday, you'll go over page 9, which has 3 additional cases that Dr. Cotter will work with you on. For the Monday group, we did those today, but if you have questions on those, just reach out to me and I can help explain them to you. But if you're not able to kind of do what I just did here, where I could take the infection going from location, determining what's causing it, and then list out first line, second line, third line responses, you're not going to do well on the exam. So this is a big piece of it. And then the rest of it will be, some of the other stuff on the case, on the study guide, review those. But I really wanted to focus down on this because if you don't study this, you're not going to do well on this exam. That is all I had. Does anybody have any questions or anything else they'd like me to go over? Um, you can type it in the chat or you can unmute. Okay, if I have nothing else, if you do have questions, feel free to reach out to me.
Um, Please,
Oh,
I do have a question.
yeah, of course,
Sorry,
go for it.
I was trying to find it, but
No, you're fine.
so on my case study, I had got -1 on the question. It asked about immune cells, list 3 immune cells, I
Hmm.
think for the allergy. I had listed, I believe, IgE, IG. Ugh, man, I'm trying to see if I had it. Give me a second. Um, 1 minute. I'm so sorry.
No, no, you're fine.
Let me just check.
While you do that, Destiny, am I able to see the other slides? What slides are you referring to, Destiny, by any chance? Or did you join late? Is that why you're asking? Okay. I'll go back and look. You can look at some of the other ones. Dr. Cotter will share this link. This is being recorded, so she will share this link with the Wednesday group after I send it to her tonight. But just to kind of quickly recap, it was just the overview, it was the details of the exam, and then just first line defense and how I have it broken down, resident microbes, and this is from your notes, from your handouts. And just like I mentioned earlier, make sure you have at least recognize 2 per location. Um, that way, if you get asked, you can either give 1 to get partial credit, or they might only be asking for 1, and you're not relying on memorizing just the 1. Second line cells, and then I went over the third line defense and then the 2 examples. But I would say, Destiny, watch the video earlier so you can get the whole scoop of it, especially since you came in a little bit late. And Fahima, did you find your question?
Yeah, I did. All right. So it said list 3 immune chemicals such as perforins, granzymes, or antibodies. So my answer had put IgG, IgA, and IgE, but she stated I didn't understand because, like, I seen you today, but since she was
Hmm.
working, I didn't really have time to ask her. So I'm
Hmm.
trying to understand what was wrong on the answer because it asked to list 3, but she said don't forget about IgM, and also that I think she said it makes IgE instead of IgG or something like that. So I'm so confused on what the answer is because what was wrong with
You
it?
said it was the allergic response. Is that what
Right.
you
It's
mentioned?
never
So
mentioned
allergic responses, because I heard you say IgG. IgG is not produced during allergic responses.
Okay.
That's on page 24 of your handout. It
Uh-huh.
mentions if you have an allergic response, IgE gets produced instead of IgG,
Okay.
and that's probably where the point came off of.
So
If you gave
I had IgE, I had IgA, and the wrong one, I guess, is IgG.
correct. So it would have still made IgM, and then instead of IgG, you have IgE. So kind of actually, it's kind of looks like it'll look very similar to
So
um,
the answer would have been IgM, IgA, and IgE.
correct for an
Okay.
allergic response. Correct.
Gotcha. Gotcha.
So that's
Gotcha.
probably where your 1 point came off of.
Right. I understand
Yep.
it now.
Yep.
But that's my only question. Thank you so much.
Okay, not a problem. And then again, if you all have any questions, feel free to reach out. For the Wednesday group, either reach out to Dr. Cotter or maybe ask her to forward some questions to me. Other than that, thank you very much for your time. Good luck on your exam and enjoy the holidays. I'll see you all next week.
Thank you. Have a good one.
Yep, you too.
Thank you.